Hi students, it's Professor Bridget, and today we are taking a step back to chapter 3. And remember that chapter 3 in it explains all of the different tissue types in the body. We didn't cover all of the tissues all at once. So far we've just covered connective tissue. So today we're taking a step back to chapter 3 to learn about the second tissue type you find in the body, which is called epithelial tissue. So as a reminder, let's look at our hierarchy of organization. We know that atoms come together to make molecules. Right? We know that atoms come together to make molecules. Molecules come together to create the organelles, the little organs inside of a cell. And those come together to create a fully functioning working cell. And that cells come together to make tissues. So far, we've covered connective tissue, and today we're going to cover epithelial tissue. And the reason we're doing it in this order is because the next lectures, the next major system we cover is the skin, or what we call the integumentary system. And that's a combination of connective tissue and epithelial tissue. So it kind of makes sense that we're covering things in this order. So today, just the epithelial tissue components of chapter 3. So let's start out looking at some of the characteristics of epithelial tissue, just kind of in general. And then as we move towards or into learning about the different types of epithelial tissue, each epithelial tissue will have a very specific structure, function, and location. So just in general, as far as where we find epithelial tissues, we find them lining the surface of the body. So if you could touch the skin on your arm, for instance, you are touching epithelial tissue. Epithelial tissue also lines all body cavities and body tubes. So think of your body tube from mouth to anus. That's all your GI tract or your gastrointestinal tract. All of that is lined with epithelial tissue. If you were to take the heart and the lungs out of the thoracic cavity, what you would find is it would be lined with epithelial tissue. And epithelial tissues are very important components of membranes. And just by definition, a membrane is a combination of two tissues: connective tissues that I'll just abbreviate CT, and epithelial tissue. So all body tubes, all surfaces of the outer body, all entrances to the body are going to be lined in epithelial tissue. Another characteristic of epithelial tissue, which is very different than what we saw in connective tissue, is the cells are very tightly packed together. So in connective tissue, we saw, for instance, like if these were fibroblasts or even chondrocytes or osteocytes, they're all kind of scattered. And in between, you'd find some fibers. So the cells weren't really densely packed, and they definitely were not connected to one another. But now with epithelial tissue, what you're going to find is you're going to have one cell very close to the next, connected to one another, and then connected to another, and connected to another, and so on. And their connections, the types of connections that hold them together, are tight junctions, and also, in some cases, desmosomes. If you don't remember what those connections are, revisit chapter 2, and those connections are explained there for you. I'll explain them too when we move on and when it becomes a little bit more relevant. And then also epithelial tissue and the cells themselves have some very distinct ends to them, or they have a distinct orientation. that we call the apical surface or the basal surface. When we get to this slide, I'll be more specific about that. It's got a picture on that next slide that I'd like to explain the orientation. Functions of epithelial tissue are going to vary depending on the structure of the tissue and the location of the tissue. In general, we can say that epithelial tissue offers protection. Some types of epithelial tissue are used for absorption. Some are used for some secretion. Some perform sensation. So again, the functions of epithelial tissue will vary depending on a couple of factors. And I'll get more specific about this here in a minute. The nutrient and blood supply of epithelial tissue is via diffusion. Epithelial tissue is avascular, meaning it doesn't have a direct blood supply. So I'll show you in a minute, but nutrients and oxygen have to diffuse to the epithelial tissue via the underlying loose connective tissue. Loose connective tissue is very well vascularized, and epithelial tissue is not. So the nutrients and oxygen and waste being removed, all that has to occur through the blood vessels in the underlying connective tissue. I'll show you in a sec. And then epithelial tissue is capable of very rapid mitosis. And mitosis is the growth and division of cells that is used to reproduce the cells, but also to help repair. So let's say, for instance, this should be review from your prerequisite course, but let's say we have a cell, right? And then its nucleus. And when an organism needs to grow, what will happen is that cell will grow in size, double its chromosomes, and then split down the center, and then it becomes two separate cells. This kind of look like boobs, I'm sorry. And then what happens is those two individual cells split in half, and then you've got 4 cells. So that's mitosis. Epithelial cells, and therefore epithelial tissue, is capable of very rapid mitosis. And usually this is for repair purposes. But again, I'll talk a little bit more about it when we get to a point that's relevant. So I want to expand a little bit on the surfaces of epithelial tissue and also how these cells are attached to one another. So if we look at the top picture, let's understand the surfaces first. So this would be the apical surface of the tissue, and this would be the basal surface. And you're probably like, well, Bridget, how do I know the difference? Well, let's consider where we find epithelial tissue. So the apical surface is going to be the exposed surface. So if we're talking about the lining of the stomach, for instance, which is a body tube or a body cavity, food would pass along this top surface of the cells. Or if you touch the skin on your arm again, you're touching the surface of these cells, the apical surface of those cells. The basal surface is the point of attachment of those cells to the rest of the body. So in yellow, what your authors are drawing right here, that's a layer of loose connective tissue. And that layer of loose connective tissue is anchoring the basal surface of these cells to the rest of the body. So underneath even that loose connective tissue, let's say that there's some muscle down there, right? So the basal surface is the surface of attachment to the rest of the body. The apical surface is the exposed surface. It's either exposed to the external environment or it's exposed to the body cavity. And then the other thing I'd like to expand on are the intercellular connections. How are these cells anchored to one another? There can either be tight junctions, like I mentioned, or desmosomes, and both of them really are proteins that anchor the cells to one another. I'll use black, and I'll just outline one cell for you. This is just one cell. And then you can see immediately adjacent to it is another cell. So the cells can't just kind of sit right next to one another and hope that they stay adhered to one another. There are proteins that are going to anchor these cells to one another. So the proteins begin inside one cell, expand or extend through the membrane, and anchor on the other side. So what this does is it holds the cells very tightly to one another, and it prevents things from passing between cells. It prevents cells from becoming detached from one another. So let's go back to the stomach example. Let's consider stomach acid, which has a really low pH, somewhere around 1.5. That stomach acid accumulates on the apical surface of these cells, and it's essentially hydrochloric acid. It's pretty gnarly. And what we don't want to happen is that hydrochloric acid to leak between the cells and one, cause damage, but two, cause the cells to come apart from one another. So these tight junctions, what they'll do is they anchor those cells together very, very tightly, preventing those cells from coming apart under normal mechanical stress, but also preventing, could be acid from the stomach or pathogens from getting between these cells and making their way into this connective tissue here. That's something we did not see in connective tissue. And then if we look a little bit more closely at the site of attachment on the basal surface of the cell, what your authors are saying is, okay, we're going to look at this attachment site right here. And they've blown up that picture for you. I want to show you how these epithelial cells are anchored to that underlying loose connective tissue. In fact, it's almost always areolar connective tissue. So the way in which those cells are connected to the connective tissue below, the connective tissue is here, right? That's the areolar connective tissue. The cells are anchored by what we call a basal lamina. And so that's this bluish structure that you see here. And the basal lamina has two layers to it. Let me just expand this so it's easier to see. So the first layer is what we call lamina lucida. I'll just write a 1 there. Lamina lucida is like an acellular glue. It's a chemical glue. There are no fibers, or few fibers, and there are no cells here. And that lamina lucida is anchored to this next layer, which is called lamina densa. And lamina densa does have fibers in it. So all of those striations that you see are fibers. So the lamina lucida plus the lamina densa are anchoring the basal surface of those epithelial cells to this areolar connective tissue down here. That's the function of that basal lamina. Another thing I'd like to talk about before we learn about the different types of epithelial tissue are some of the specializations that we see on the apical surface of epithelial cells. So again, just as review, here's one individual cell. And you see a few here, right? Here's another. Some epithelial cells will have specializations on their surface. Some will not. I'm not suggesting that all epithelial cells have specializations, but often you do see specializations. So let's look at the shortest of these little projections first. On this cell right here, you see really tiny projections all over, just almost like little goosebumps all over. Those are called microvilli, this first one. Microvilli are extensions of the plasma membrane or these kind of like bumps in the plasma membrane that help to increase the surface area for absorption. So I'll write it here. Increase surface area for absorption. So we normally find microvilli on the surface of epithelial cells that are performing the function of absorption. So let's say this epithelial cell would like to absorb nutrients. Maybe it's one of the epithelial cells that lines the small intestine. So nutrients are passing over the surface of this cell, and the more little projections that are coming out of the cell, the more nutrients can be taken into the cell. You're increasing the surface area for absorption to occur. Next, we'll move to cilia, which are just a little bit longer than microvilli. And that's what you see here, these longer projections. Cilia do not increase the surface area for absorption. Cilia have the ability to move back and forth. Well, I shouldn't say back and forth. They move in just one direction, so we'll just say forth. What they do is they have the ability to bend all at the same time. So let's just say all of these projections right here are going to bend to the right all at the same time. And the function of those cilia is to move fluids across the cell surface. So it's one way in which we can propel substances from one cell to the next, to the next, to the next, right across the surface. And then another specialization with a similar name but a very different function are the stereocilia, and they're much longer. These structures do not have the ability to beat or move on their own, but if something comes in contact with them, they do bend. And that sets off a reaction inside of the cell that cell is often connected to a nerve cell. And so it causes changes inside that nerve cell just by moving those stereocilia. And then this little bolt of electricity, or this signal that we call an action potential, gets sent down that nerve to the brain, ultimately. So these stereocilia would get bent. That causes an internal change inside the cell. That internal change inside the epithelial cell then causes a change inside the nerve cell. And then the nerve cell kind of fires off this electrical signal back to the brain. So stereocilia are going to be found on sensory types of cells, like the cells in your inner ear. Okay, so let's move on to some specifics. Let's first look at how epithelial tissue is classified. How do we organize epithelial tissue? And there are two ways. We name epithelial tissue based on the number of layers of cells in the tissue and based on the shapes of the cells. So for instance, a simple epithelium is just going to have one single layer of cells. And then beneath that, you would have some areolar connective tissue, right, on that basal side. A stratified epithelium, and that is misspelled, there should just be one I. A stratified epithelium is going to have more than one layer. So strata means layer, right? So I'm just going to draw in a tissue that has 3 layers, for example. And then pseudostratified, I'm going to show you in a picture because it's kind of hard to understand without looking at a picture. But know that pseudo means false and stratified means layered. So this is falsely layered. It appears as though there are multiple layers in the cell, or in the tissue rather, but there really is only one true layer. And then the other way that we classify epithelial tissue is based on the shapes of the cells. So squamous, as you may have learned when you were learning about the skeletal system, Squamous actually means in Latin scale-like. And what they're referring to is fish scales. So if you were to look at an epithelial cell kind of from the top, it kind of has a shape of a fish scale. And I don't expect you guys to know what that looks like. But it kind of looks like, it kind of looks like if you were to fry an egg in a pan, if you're looking down on the cell. So squamous is going to have this very thin shape. So if you're looking down on that cell, it's like you're looking down at a fried egg in a pan. But if you were to look at that cell from a side, it would be real thin. There'd be a little bump like your yolk, right? And that yolk would be the nucleus. So these are the thinnest of cells, the narrowest of cells. And there's a reason for that, right? Structure function. And then cuboidal, pretty easy. The term is pretty explanatory. It's just going to have a cube shape. And the defining feature of cuboidal cells is that they've got this really big nucleus in the center that pretty much occupies the majority of the cell. Columnar epithelial tissue cells are, again, as the name implies, they're taller than they are wide. And what often defines these cells is that they have a nucleus that's closer towards the basal surface than the apical surface. And then lastly, transitional epithelium here. I'm going to come back to this because, again, that is kind of like pseudostratified, where it's difficult to really understand unless you're looking right at a picture. So we almost always put these two classification names together when we're naming an epithelial tissue. So you might have what's called simple squamous, or there is stratified cuboidal, or simple columnar. Almost always we use two words to describe the tissue. The exception being the oddballs, pseudo-stratified, and transitional. And I'll, again, show you examples of that when we get there. So let's look at squamous epithelium first, both simple, one-layered, and stratified, multi-layered. So again, let's look at the shape. And they really do. When you go to look at the histology, these cells really do kind of look like fried eggs, where, again, you've got the nucleus right there in the center. And in this picture, they are kind of showing you what it would look like from the side, too, where you have your yolk, your nucleus right there, and then, like, all of this would be the egg white. Egg white, sorry. I'm distracted. My cat's laying on my keyboard. Okay, so in this case, this is simple squamous epithelial tissue, meaning there's just one layer of cells in the tissue, and immediately deep to that is going to be that connective tissue that anchors that epithelial tissue to the rest of the body. Where we find tissue like this, simple squamous epithelium, are in areas where diffusion is occurring or secretion. So diffusion, meaning the exchange of something, usually into the cell, and secretion would mean secreting something out of the cell. So I'll give you a couple of examples of where we find simple squamous in the body. One is the capillaries. Capillaries are the smallest of your blood vessels, and this is where, let me see if I can draw a capillary here. So here's a capillary in cross-section, and let's say its walls are made up of simple squamous. So these little lumps are the simple squamous, right? So what's being carried in your blood? Well, there's oxygen, there's glucose, I'll just write those two in because I'm running out of space. But there would be tissue surrounding that capillary, live tissue, live cells, right? And those live cells are going to require oxygen and nutrients like glucose. So in a capillary, you've got these, the simple squamous that make up the wall of the capillary, so that oxygen and glucose can easily diffuse out of the blood vessel and into the surrounding tissue. So the thinner the wall of this capillary is, the easier these molecules diffuse out. And in addition to that, and this is kind of getting ahead of ourselves, on the outside is going to be waste products like CO2. And we want to get rid of CO2, right? So the CO2 outside of the cell can diffuse into the cell and continue on with the blood away from the tissue. So that's an example of how simple squamous epithelial tissue is good for diffusion. Another example, it would be the alveoli of the lungs. And let me see if I can draw one of those. I'm not sure. So alveoli kind of look like little grapes. And this is where gas exchange occurs. So if you take a deep breath in, all the way down, what's coming in as you take that breath in the air that you breathe is oxygen. And those alveoli are lined with simple squamous cells. And the oxygen isn't going to do our bodies any good just sitting in that little alveolus. Alveolus is singular, alveoli is plural. Oxygen's not going to do us any good there. What needs to happen is that oxygen needs to diffuse out of the alveolus and into a capillary. And then that capillary carries that oxygen all throughout the body. So it's no coincidence then that both an alveolus and a capillary have the thinnest cells possible making up those structures, because diffusion is really hard through very thick tissues. So we want very thin layers of tissues for diffusion to occur quite easily. So that's simple squamous epithelial tissue. And then stratified squamous is layered. Remember, stratified means layered. So in the picture below, all the cells that you see in pink here, all of those are the stratified squamous. And one of the things you'll notice real quick is that all those cells there, they don't all appear squamous in shape. You're like, Bridget, well, you told me they're all going to look like fried eggs. Well, here's the deal with stratified squamous, is that these cells here at the apical surface of the tissue, they do have that squamous shape. So when you're trying to identify the tissue that you're looking at, if it's a stratified cuboidal or a stratified columnar or a stratified squamous, always look towards the apical surface of the tissue, because these cells down here have not taken on their adult shape yet. These cells are new cells. And the way that this works is this layer of cells down here is the layer of cells that's constantly growing and dividing. The layer of cells that's undergoing mitosis constantly. That's the growing layer. So those cells right there, they grow and divide. They're going to split in two, right? And then one of those cells moves up a layer. And then when the next cell undergoes mitosis, it moves up as well, and it starts pushing all of these other cells towards the apical surface of the tissue. Kind of like a little escalator of epithelial tissue. And so these cells don't really take on their squamous shape until they get far enough away from where they began. And let me tell you the reason for this. In this connective tissue down here in yellow is where you're going to find the blood vessels. Remember, all epithelial tissue is avascular. And so any nutrients that the cells need are going to leak out of these capillaries and diffuse to these cells. These cells that are most important. These cells right here are the cells that are carrying out mitosis. They're the ones that need oxygen and nutrients. And as these cells grow and divide, and as these cells get further away from the growing layer, they get further away from their blood supply. They get further away from their nutrient supply. So essentially, they're dying as they move away from that growing layer, this growing layer right here. So as the cells move away in this direction, they're getting deader, so to speak. So then they start to kind of dry up a little bit and take on that squamous shape. Okay, so generally the function of stratified squamous is protection. Protection from abrasion. Let's say, for instance, you burn your mouth on hot coffee. Let me erase some of this. You burn your mouth on hot coffee, and you damage maybe the first 3 layers of cells. Well, you've got stratified squamous in your mouth, and so that hot coffee may just only damage a couple of these layers of cells, right? So not a big deal. There's still plenty of epithelial cells there, right? To still create that lining of your mouth. But more importantly, let me erase all this. More importantly, think about pathogens. Pathogens, microorganisms are constantly trying to invade our bodies, right? And how do they get in? They get in through scratches or cuts in the skin. They get in through the mouth, the anus, the vagina, the nostrils, anywhere they can find the respiratory tract. They're going to try and set up inside the body in a favorable environment. So let me go back to talking about the blood vessels here. Remember, all epithelial tissue is avascular, but there are blood vessels down in here, right? In that loose connective tissue. Having many layers of epithelial tissue is going to create a barrier between the outside environment where there might be, I know what I can draw. Might be, and this is not how this occurs, but I'm just going to draw a little COVID with its spike, right? It doesn't come in through the skin that we know of, but and it would be much smaller than that. But we don't want those microorganisms to pass through that epithelial tissue and then get into the loose connective tissue, because then what can happen? Those microorganisms, they make their way into the blood vessels, and then it's like a freeway through or an interstate through the body where they can go and wreak havoc in the body. So having multiple layers of cells provides a nice thick barrier to entry of microorganisms, but also to if some of these layers get damaged via abrasion, like maybe you even eat something that's really abrasive, no worries. There's still plenty of layers of cells there to protect this soft connective tissue underneath that has those blood vessels in it. So examples of where we would find stratified squamous: the epidermis, which is your outer skin that you can touch. That's a really important barrier. The mouth, the inside of the mouth. So if you take your finger and kind of scrape inside of your mouth, or if you've ever done a DNA test and you've swabbed the inside of your mouth, what you're collecting are those epithelial cells, just a few from the surface. The esophagus, the tube that carries food and drink down your stomach, is made of stratified squamous. The anus, which is an entrance to the body, right? The vagina, which is an entrance to the body. All body entrances are going to be made of stratified, I shouldn't say all. Most body entrances are going to be made of stratified squamous. Again, because microorganisms are trying their best to come in and set up shop in our bodies. So this is our body's protective barrier. Now I say that stratified squamous functions in protection, but even stratified cuboidal or stratified columnar are going to have a protective function as well. It's not just stratified squamous. Just want to make that clear. Okay, so here's an example. If we were looking down on squamous, we can't tell if it's stratified or simple because you're looking down on it. I can kind of tell that it's stratified, but if you haven't seen this tissue before, you wouldn't know that. But the reason I'm showing you this is I really want you to see the shape of the cells. And there is only one nucleus here, and that's here. This is a nucleus from a cell that's below it. So they really do look like fried eggs. And you see how they are very densely packed, and they're going to be anchored to one another. Oh, so this would be like looking down at the skin, for example, the epidermis. And here's another view of simple squamous, but you're looking at those cells from the side. So they look more like fried eggs. If you were to look at your fried egg from the side, or if you put your fried egg in a sandwich, it would look like more like looking at that fried egg between the two pieces of bread. Let me outline one of these cells for you. Let's use black. So here's one of those simple squamous cells. All of these are simple squamous, but they're not all obvious because there's a lot more going on here. But imagine these squamous cells kind of all lined up end to end, right? And this entire structure right here, this entire structure is an alveolus. That's the structure that carries out gas exchange. That's the terminal or the very end point of your respiratory tract. So here, as you take a breath in, oxygen is going to make its way into that alveolus, and they're going to be teeny tiny little blood vessels. Let me see if I can find one. Looks like there's one right here. Teeny tiny little capillaries that are also made of simple squamous that are going to be able to allow that oxygen to diffuse into your bloodstream, and the oxygen then gets carried throughout the body. Here's an example of stratified squamous. Let me write that. And there are two types of stratified squamous. Let me show you what it is first, and then I'll show you about the two types. This really isn't all that relevant until we get to the skin. But you're looking at your skin here underneath a microscope. All of this, from arrow tip to arrow tip, all of this is stratified squamous. If we were actually looking at this underneath the microscope, we could zoom in and we'd have much better resolution, and you could see that those cells are squamous in shape. That's something that you'll be doing for lab. But that's all stratified squamous. And what is all this? Can you guys identify this type of tissue? It's something we've seen before. I'll give you a couple of seconds. And hopefully you all said dense, irregular connective tissue. You've seen that before, right? Okay. So this is what we call keratinized stratified squamous, also known as dry stratified squamous. And let me tell you why. This layer right here that looks a little flaky, those cells are full of a protein called keratin. So that's why we call this keratinized stratified squamous. These epithelial cells produce that protein called keratin, and it makes a really waxy protective layer. So go ahead and look at some of your hair products, or go and look at your lotions, and you'll probably see on the front, or maybe in the back on the ingredients, keratin as an ingredient. Keratin is a waxy protein. So in order to moisturize your skin and prevent skin from leaking more water, which would that water would come from the connective tissue down here, you put a waxy substance on the surface, and that holds the water in, holds moisture in. But this layer right here that you see that I'm bracketing in green, those are squamous cells, several layers, so that's stratified squamous, but they're filled with that protein called keratin, and it makes them really kind of flaky and gives the tissue this very distinct appearance. Now these cells down here are also stratified squamous, but they're not filled with keratin, not yet. Let's move on to this type of stratified squamous would be the stratified squamous that you find on your skin. Okay, so skin needs to have that waterproofing barrier. We don't want to lose our water to the external environment, so we've got this nice waxy layer. Keratinized or wet stratified squamous. So here's an example where you don't see the flaky layer of cells on top like we did in the previous picture, right? You don't see that. That's because these stratified squamous cells do not produce keratin. So you don't have that flaky, waxy layer. From arrow tip to arrow tip, or bracket end to bracket end, this is all stratified squamous. But these cells don't make keratin. They don't have that dry, flaky layer. So what's the point then, right? Well, a non-keratinized or a wet epithelium would be found on what we refer to as mucous membranes or wet membranes. So this type of tissue could be found inside your mouth or lining the vagina. But they're both stratified squamous nonetheless. So let me tell you a little story about stratified squamous epithelial tissue. I said that one of the places that you can find it is the vagina, but you can also find it lining the cervix. So if you've ever gone in for a pap smear, or if you don't get pap smears yourself, you know somebody who does go in to get regular pap smears, the OBGYN or the PA that's doing the procedure will take a device and scrape off some of the cells of the cervix. The cervix, as I mentioned, is lined with stratified squamous. And what the doctors are checking for are any abnormalities in these stratified squamous cells, any visual abnormalities, which could indicate cervical cancer. Remember, one of the characteristics of epithelial tissue is rapid mitosis, rapid cell growth. So if one cell, for instance, acquires a mutation that could potentially be cancerous, then if that cell grows and divides, it passes on that mutation to another. And then that cell also grows and divides. And so you have all these cells then, potentially, that have a mutation that's cancerous, growing and dividing, growing, dividing, growing, and dividing, which can occur really, really quickly. And cervical cancer can be treated if caught early. And with cervical cancer, it's normally caused by human papilloma virus, called, or often abbreviated, HPV. And if you work in healthcare, you probably already know this, but it's estimated that 90% of human, I don't know if it's all humans, but let's just say Americans, both males and females, are carrying HPV. And males are much less often to experience symptoms of HPV or complications of carrying it. It's kind of a silent virus. For women, when they're first infected, they may develop a slight fever, or may not even be ill at all, may not even notice. But over time, this virus causes a cancerous mutation in these cells, and these cells grow and divide, grow and divide, grow and divide. And when those cells escape the cervix, they can get into the blood vessels, right? When they get into the blood vessels, those cancerous cells can be transported anywhere in the body and create cancers elsewhere. This is what we know as metastasis, or this is how cancer spreads, gets into the blood vessels. And so it's important that you or your loved ones go in and get regular pap smears, especially when you're young, and be tested for HPV. Because HPV is the number one cause of cervical cancer in women. And it is can be lethal if it's not treated. If it's left to metastasize, it can kill. But if it's treated early, it can be quite benign. What's pretty neat now is that in the last, 10 years, there's now an HPV vaccine. This is the first time in human history with the development of this vaccine that we have actually got away a solid method of prevention of cancer. There are a lot of ways in which we can treat cancer, and we can advise individuals not to smoke or not to drink too much or to avoid specific lifestyle choices in order to help to decrease the odds of developing cancer. But the HPV vaccine is the first time in history that we've been able to prevent cervical cancer, prevent a cancer. So it's a huge win for medicine and women's health. Let me tell you about Henrietta Lacks. Henrietta Lacks was a woman who was living in the 1950s and was experiencing some issues and went to see her gynecologist. And he did a pap smear, looked at the cells underneath the microscope, and saw something very unusual. He saw that these cells had acquired a mutation, so she definitely had cervical cancer. And what he did with those cells, this doctor, was he tried to grow them in a petri dish. At this time, in the 1950s, we were able to keep cells alive in a petri dish, but we couldn't get them to reproduce in a petri dish. Not easily, anyways, not for several generations. But Henrietta Lacks' cells grew like crazy in a petri dish. So much so that the doctor started giving away those cells to other doctors for research. And over time, her cells, which were they were cancerous, right? But they were so good at growing and dividing and do it so readily in a petri dish that they were being used to test and do research on cancer. All of this without Henrietta's knowledge. She succumbed to her illness a few years after her diagnosis, but permission to 1, harvest her cells, and 2, grow them in a lab, and 3, distribute them all over the world so scientists could do research on those cells was all done without her permission, without her knowledge. And this is kind of common in the day, but the important thing now is that her cells are still alive. They're still alive in labs all over the world, and they are now referred to as what we call HeLa cells. And they're used for cancer research and cancer treatment research. And so imagine all of the money that drug companies and major corporations have made off of her cells in developing this drug. And had she been alive now today to see this, I'm sure she'd be quite proud. But she wasn't told. Her family wasn't told until several years later. Now, ultimately, those HeLa cells, her cells are of course helping with research into cancers. But finally, now her family is getting compensation for all of the strides made in cancer research, which is good. A little late, but good. So let's move on to the second shape of epithelial cells called cuboidal. And as you know, there can be simple cuboidal or stratified cuboidal. Remember that cuboidal cell is cube in shape, or cuboidal in shape, I should say. And one of the defining features is that it's got this giant nucleus in the center that occupies the majority of the cell. Cuboidal epithelium, whether it's stratified or simple, is usually used for either secretion or absorption, sometimes both. So if we're looking at, for instance, one of the tiny tubes in your kidneys are used to filter your blood, and that blood filtrate, what's filtered from your blood, contains molecules that your body might want to reabsorb, and we might want to secrete molecules into that fluid. So let's say, for instance, that filtrate is passing in this direction. You're looking at a sagittal section down the tube, and let's say there's fluid in here. It's not urine yet, but it will become urine. But in that fluid, there's some sodium ions and there's some chloride ions, right? And your body's like, wait a second, I could really use the sodium and chloride ions. Let me reabsorb those. And so those things can get reabsorbed into those cuboidal cells. So your body hangs on to those things. Those ions don't get lost in urine. One of the other things that your kidneys do is to help to regulate the pH of your blood. pH, or the acidity of your blood, is dependent on hydrogen ions. So we might want to secrete some hydrogen ions into the filtrate. So those cuboidal cells can act in either secretion or absorption. Obviously, here's the drawn picture. Here's what it actually looks like. Those kidney tubules underneath the microscope. Stratified cuboidal is usually just found in sweat glands, and they usually just function in secretion. So they're going to secrete that salty sweat that we know, or even the stinky sweat. So if we're looking at a gland in cross section, that's the part of the gland, and then here's the tube that would lead to the surface of your skin. Here's a cuboidal cell, and here's a cuboidal cell. And so in this case, in the case of a sweat gland, the water or salty, watery product that we know as sweat is going to be secreted into that duct right there and taken to the surface. Okay, so let's look at the types of columnar epithelial tissue. Now here there is simple, like we've had before, and stratified. But now we see this weird long named one that we haven't seen yet. So I'm going to cover simple and stratified first, and then I'll cover the pseudo-stratified. So simple columnar epithelial tissue is going to be one single layer of cells, right? And remember, the thing with the nuclei of columnar cells is that they're closer to the basal surface than they are the apical surface. So the nuclei kind of line up in the same plane. So you could almost draw a straight line right through the middle of those nuclei. That's one characteristic of simple columnar epithelial tissue. You can see that these cells right here have microvilli on their surface. Right, I'll just draw them, emphasize them. So what that tells you about the function of this tissue is that the tissue is designed for absorption. Remember, microvilli increase the surface area for absorption. And so if this were the small intestines, for example, those nutrients that you've ingested would get absorbed into these cells and then taken down through those cells, and those nutrients end up in blood vessels that are in the connective tissue, and those nutrients then get carried away to the rest of the body. Stratified columnar is kind of as the name implies, really, in that you've got more than one layer of cells. So here you go, right? I don't know why they didn't draw a nucleus in this one. I'll draw it in there. And there's another one, then another one. So stratified columnar, you've got these columnar-shaped cells, more than one layer. They're primarily for protection. Remember, we've got that double layer of cells, so any stratified epithelium is good at protection. And these cells are going to be secreting substances. So where we find them, location, a salivary gland, so those saliva glands in your mouth, or even the urethra. The urethra is the tube that carries urine from the body to the external environment. So in that case, they're protecting the urethra. In salivary glands, they are secreting substances. So let's move up to the middle of the slide, and let's look at this tissue called pseudo-stratified ciliated columnar epithelial tissue. It's a mouthful, I know. Pseudo-stratified. Remember, pseudo means false. And so if we look at this tissue, it appears as though there's more than one layer of cells. You've got a cell right here, but then you've got a cell right here. And so it's falsely stratified in that each one of these cells still has a connection to the basal lamina. Here's the basal lamina. I'll draw it in green. Here's the basal lamina. And so each one of these cells still has a connection to that basal lamina. So here's a connection for this cell, right? And then here's the connection for this cell. And here's the connection for this cell right here, right? So they're all still maintaining their connection to this basal lamina and then the underlying connective tissue, right? Whereas if we look at truly stratified, let me erase some of this, a truly stratified epithelium, these cells right here, the top cells on the apical surface, they're not maintaining a connection to that basal lamina down here. These cells on top, they're connected immediately to the cell beneath them. So they don't maintain that connection to that basal lamina. So that would be truly stratified. Where we find pseudostratified ciliated columnar is in the trachea, what you might call your windpipe, the tube that's carrying air from your nose and mouth down to your lungs. And there, the pseudostratified ciliated columnar epithelial tissue function as protection, right? So this is not a simple layer of epithelium. So even though it's pseudostratified, it's still pretty thick, right? So it offers protection. And then also, what you don't see in this picture, there would be little cells embedded between the columnar cells here. And those would be mucus cells. So those cells are going to produce mucus, and that mucus gets onto the apical surface of the tissue. And that mucus is meant to trap debris, dust, pathogens, anything that you inhale. And the cilia on the surface of those cells are all going to beat in the same direction to move that mucus, plus what's ever in that mucus, up and out of your body. So think about Southern California wildfires, or if you've been near a wildfire with where the air quality is poor enough that you feel it, that you recognize it. And maybe you've been outside and for some period of time, and you come in and you blow your nose. And the mucus coming out of your nose isn't a clear mucus, but it's the color of the ash or whatever was in the air. And the reason that you see that ash entrapped in that mucus is because of this type of tissue. It's entrapped the debris and the ash, and the cilia have beat that mucus up and out of your respiratory tract in order to protect your lower respiratory tract. The same is true with viruses or bacteria. The mucus there should trap viruses and bacteria and bring them up and out of the body rather than allowing those viruses and bacteria to take hold deep inside your body and cause severe infection. Okay, and here's the last type of epithelial tissue we're going to learn about. This is called transitional epithelium. And notice here how we don't use the term simple or stratified, or even pseudostratified for that matter. It's just called transitional epithelium. And let's talk about the shape first. Transitional epithelium can take on different shapes. And the reason for that is the cells are really tightly attached to one another. Let's have a look here. The cells are really tightly attached to one another. Remember, you're going to have those tight junctions attaching those cells together? But these cells are extra tightly attached to one another. And so you find these epithelial cells in organs that need to stretch. So think of the bladder, for instance. As urine fills up the bladder stretches to accommodate that urine. And those cells, when the bladder stretches, they're not going to come apart from one another. But what will happen is they become a lot wider. So let's look down here. This was an empty bladder. So you can see how tall those cells are. But once that bladder has been filled up, you see the cells now have become a lot wider. They still haven't dislodged or detached from one another, right? So they're able to stretch. And since they're able to stretch, they don't always have exactly the same shape. So we don't call them squamous, we don't call them cuboidal, we don't call them columnar. They can exist in transitional forms, so to speak. So many students will say, well, how do I tell the difference between transitional epithelium and stratified squamous, for instance? And it's always about looking at the apical surface of the tissue, the side of the tissue that's closest to the inner lining of the organ or the surface of the body. And you look at the shape of the cells. And you don't see a pattern in the shape. They're definitely not squamous. They're definitely not cuboidal in shape. They may be kind of almost considered columnar, but look at the position of that nucleus. If this were columnar epithelium, that nucleus would be way down here, right? So transitional epithelium doesn't look like any of the others. You cannot find a pattern for the shapes of the cells, whereas in the others, you definitely can. All right, and the last slide I've put at the end of part 1 here is a nice little chart. I love charts. I love being able to organize information in a chart form so that it's easy to access and you get straight to the point, you get straight to the facts. And so I encourage you guys to do this every time we have a new system or whenever you're struggling with a lot of information, see if you can organize it into a chart. So this chart shows you a picture of the tissue on the left. It shows you an example of where you find that tissue or examples of where you find that tissue and functions of that tissue. This is from a textbook. I don't think it's from our textbook. If any of this varies from what I said, please let me know. I may not have said it in exactly the same way, but all of this is accurate information. So here is one whole lecture in a chart, just like we had for connective tissue. You can compare visually the structure of the tissues, compare the location of the tissues, and quickly compare the functions of those tissues. Okay? That's all I have for part 1. Coming up is part 2 of epithelial tissue, which, if it makes you feel any better, is a lot shorter than part 1. Okay, see you soon.